Hi, my name is Shreya and Narayan, and I'm going to be talking about the inhibition of receptor cluster and recognition in anaphylactic reactions. My own experience with anaphylaxis drove me to do some research in this. I wanted to prevent reactions from occurring as opposed to taking a reactive response and taking medication after an allergen has been ingested. So, the anaphylactic response occurs because of mutations in genes of the immune system. Essentially, there is false recognition of non-harmful particles as harmful. These antibodies called IgE antibodies are produced. They attach to mast cells and release histamine, which causes all the symptoms. So the idea I have is to inhibit the production of histamine and prevent the amount of receptor clustering that will be required to instigate a response. So parallels can be drawn between this macrophage activation and between mast cell activation. In macrophage activation, or in the process of phagocytosis, an appropriate amount of receptor clustering is required for an actual response to occur. So parallels can be drawn with mast cells in that they only respond with the binding of IgE antibodies. And once that binding occurs, and once that amount of clustering actually happens, histamine is released and the reaction has started inside the body. So the idea I have is to change, or in this case, increase the amount of receptor clustering that will be required for an immune response to occur. So essentially, although the body would still produce antibodies to wrongfully target allergens inside the body that have been ingested, this reaction that would be potentially intended mistakenly by the body would not occur because enough receptor clustering of the IgE mast cell complex would not happen for the histamine to actually be released. And so we would be increasing the amount of receptor clustering that we required so that, in effect, no release of histamine actually occurs, or a very minimal amount, so that reactions are lessened. So inhibition can be achieved through CSKs, which generally have been shown to inhibit SFKs, or Sarc family tyrosine kinases. The CSKs are negative regulators of SFKs and can be used to prevent this kind of phosphorylation of the SFKs in these processes. The only problem that would be needing some additional research is that we don't want to target the necessary processes of SFKs, such as regulation of cell growth. So we'd want to make sure we only target the part of the SFKs that is allowing for histamine release to occur. So we want to specialize in our target of inhibition. Other inhibitors that we can use instead of CSKs include a plant called Lindera obtusilova, found in Eastern Asia, of the Laraceae family, which has been shown to be involved in histamine regulation as well. So this is a brief synopsis of the research that I have done in combating anaphylactic reactions.